enhancer binding proteins that bind the enhancers onto the gene. I will reiterate, the DNA is the same in every single solitary cell in your body, except the immune system. And thus, you have to have a way of regulating when a gene is on or off in a different tissue. You don't want a gene on in your red blood cells that has no place being there and can in fact destroy it. Like there are rhodopsin molecules you do not want expressed in your red blood cells, and you don't want hemoglobin expressed in your rod outer segment. You want to be able to drive specific expression and high levels of expression sometimes. So you need enhancers to drive that. And when we refer to enhancers, we refer to the proteins that bind the sequences on the genes. We recognize those sequences on the genes by these sites that we call enhancers. So, for example, if E1 protein is expressed in a particular tissue, and that tissue is legs, then this gene will be expressed in legs. If E1 and E2 are expressed in wings, then this gene now can be expressed in wings. For example, that doesn't, so I think you have a failure to realize, does that mean in wing tissue, if you have E1 expressed only, will it be expressed? No. Right? Just because it's wing tissue and just because you have E1 expression does not mean it will be on. As I said, you need E1 and E2 on in wing tissue in order to get it expressed. Even though E1 is all that's necessary to get it expressed in legs. And that's because there are other molecules in the cell that regulate along with E1 and E2 that are beyond the control, beyond this gene. There are things that are cofactors of E1 and E2 which won't allow, for example, E1 alone to turn this on in wing tissue. It's beyond the scope of this discussion. So E1 alone will not be expressed in the wing. You need E1 and E2 to be expressed in the wing. If you're E1 plus E2 plus E3, maybe you're expressed in the antenna. Because E1, E2, and E3 cooperate to drive expression of this gene in the antenna. That does not mean if you have E1, E2, E3 expression in the wing, you will necessarily get that gene on in the wing. And this is how we describe enhancers. We describe where those enhancers are functional. And they're functional based on an analysis. And we do this analysis in a very, very simple way. For example, we might delete E2 and E3 and ask, where can you get that expression of the gene? And so, for example, let's take this. This is a very important example. So if I delete E2 and E3 from the gene, where will this be expressed? In the legs, okay? Now, what if excuse me?
There are 4 enhancers.
I know, I haven't touched E4 yet. So, despite E2 not binding to the gene, it's still not expressed in the wing. Right? Just because E2 is deleted, just because E2 happens to be expressed in the tissue, does not mean that it regulates the gene. Those enhancers are specified for a given functionality. Because there are many, many other players that drive expression of a given gene. We just say these sites are necessary, and that's how they're defined. E1 is necessary for leg expression. E1, E2, E3 are necessary for wing expression. That means if we delete E1, E2, E3, we won't, or E1, E2, E3, I should say, we won't get it expressed in the antenna. If we delete E3 only expression, we'll get it in the wing and leg. It's a way of describing enhancers, and I know some of you might think it's a convoluted way of describing enhancers, but it's the only mechanism we have, because the way we do experiments and the way we think about biology is, all we have is the ability to mutate an enhancer and ask where, an enhancer site, and ask where we now express our genes. Yeah?
The way you said that just before, it made it sound like if you delete E3, you'll still get E3 in the
You still have the protein expressed in the...
Yeah, but you have the E3 protein expressed, or you get that gene expression?
It's not the E3 protein, right? Because in the wings, you never had E3 protein made.
You'll still get expression of that gene in wings and legs. You could have E3 in antenna, but it won't be able to bond, so you won't get expression of that gene.
You will not get expression of that gene in the antenna.
But
Period.
You'll still have E3 protein.
You'll still have E3 protein in existence.
And you don't need E3 for that gene to be expressed in wings and legs.
That's correct. Right? Yes.
So what you're saying is that you're mutating the E3 enhancer so the proteins for E3 don't no longer recognize it?
That's correct. Right? Remember, it's 6 nucleotides or so. If you mutate any one of those nucleotides, there'll be a crippling effect. Remember, a restriction enzyme is only, it's actually a palindrome of 3 nucleotides. Usually this is E401. It actually binds as a dimer recognition sequence. It's just 3 nucleotides. Can we have a little quiet in the room, please? It has a 3 nucleotide recognition sequence. It binds as dimer. You mutate this 1 nucleotide, that's the end of it binding. So 1 nucleotide really can be cripple protein from binding DNA. It's very, very easy to do it. Yeah?
You said it binds the dimer to 3. Each one of the 3 parts binds to a different one of 3. That's how it gets segmented.
Yes.
Okay.
Yeah. So restriction enzymes happen to bind as dimers, but that's not all transcription factors. And EQR1 is not a transcription factor. It's just it's a nuclease. Yep. There was a question.
So we're talking about juicing from tissues that are already rated as for tissues.
That's correct.
So basically what I'm saying is this gene will be, like, there's other genes where if it's expressed in light, for instance, there's another gene that's expressing that codes for the E1 protein.
Sure. That's right.
Is that what makes the tissue the tissue? Or
That you don't know unless you. I mean, there's a tremendous amount of proteins expressed in a given tissue. So I mean, you have to work out the circuitry.
so this is talking about where genes are expressed rather than how healthy.
That's correct. That's correct. Yep.
The enhancer sequences exist in every gene, in every cell.
That's correct.
Okay, so what makes the proteins not bind to enhancer free, let's say, since the protein exists in the cell?
No, it doesn't exist. You don't make E3 protein in the
That's
legs.
what you said before, no? That the protein, E3 exists, the protein exists.
E3 will still exist in antenna, but it won't be.
The
It won't have
E3
any
protein
other
will
function.
exist if I mutate the site. The protein never went away. But it can no longer bind that site, it can no longer regulate this gene, and therefore this gene will be off in antenna.
Because that specific site is mutated.
That's correct.
So in every differentiated cell, there's a mutation in that enhancer.
In every cell,
In every cell.
there was
When you say E3 protein, are you talking about transcription factor or something along the outline?
I don't hear the question. Say it again.
When you're talking about an E3 protein, are you talking about it as something like transcription factor that's
Yes.
supposed to bind to it?
Yes. So enhancer sites bind to transcription factors. That is basically the definition of them. Got it?
Are you saying that the same gene is regulated in different tissue, like by having different enhancer sites?
That's correct. That's correct. And I mean, I hate to tell you, I'm way oversimplifying this. When you get to developmental biology, this looks childish compared to the way it really works, where you have these complex interaction loops going on. This gets really, really complex, really, really fast. Yep.
Do the genes for transcription factors
Sure, how do they get on? That's exactly right.
So is there sort of an oldest transcription factor?
Is there a what?
An oldest transcription factor that sort of turns more things on?
So this goes back to how we looked at sex lethal. If you remember how we looked at sex lethal, what we saw was, and what I was trying to convey to you, was that a twofold difference in a gene expression could essentially turn off one pathway or turn on one pathway. So remember, during mitosis, you have cells that are precursors to future cells, which have to divide. As soon as they divide, they become two different identities, because you change the loops going on in those individual cells. That's part of the reason why I went through sex lethal, because you see how twofold difference in one type of gene or one set of transcription factors radically alter the entire fate of those cells. So what it is that you have early cells, they divide, the two new cells are either one is the same as the parent cell, or they're both different. So the way, for example, stem cells, when they divide into two, for example, sometimes remake the stem cell, and you make a daughter cell. That stem cell stays exactly the same. The daughter cell is missing transcription factors. Do you know how one would do this? This is sort of a real aside. Do you know how one would keep one like this and have one different? Yeah? Unequal division is essentially close, yeah?
Would you have factors that would reduce it? Actually, I'm not the
Yeah? Oh, I want to stay on this question. So the way you do it is you actually take the factors that keep it stem cell-ish and you segregate them into these cells. So you physically move the cells rather than allow diffusion-limited separation between two cells. You retain them in one of the daughter cells so that it retains stem cell-like qualities. So then the future cell is a daughter cell, and it's different. So but most cells in your body don't do that. Most cells, when they divide, make daughter 1 and daughter 2 cells. And then, of course, they now have different transcription factors expressed within them. And you see how this goes on and on. These can divide a little bit longer, make more D1s, and then it could hit another step where they're going to divide based on some extrinsic factor that now makes daughter 3 and daughter 4 cell, for example. Yep.
And how do we go from body cells to stem cells?
But stem cell is just another cell.
They have all the transcription factors.
No, remember, there's no such thing as, well, there really is no such thing as a pluripotent stem cell. So, and again, I'm really going off subject, I don't want to just yet. Go.
When we're talking about knocking out a cancer site, are we knocking out, assuming, for instance, that a cancer site is used in multiple different genes, in combination with other ones, are you knocking it out the entire genome, which is not that?
There are many ways to do that. You can knock it out in one type of cell, or you can knock it out in the entire genome. We will get to that when we finally. You can knock it out in one type of cell if you want.
But I'm getting kind of the position of like, if we're talking about one type of cell, then in that cell, it's not going to have one cancer site in any of that cell's genome.
That's correct.
So all of the genes that need that enhancer site will be.
There's only one gene that needs that enhancer site.
So the enhancer site is the same.
Yes.
Okay, so there's no such
That's part of the definition of a gene. So when we, in the first few lectures, we defined a gene. Remember how we defined a gene? It was exons, introns, promoter, enhancers. So the enhancers were part of that definition of the gene, no matter how far away the enhancers were. In fact, one of the things I told you in class was that enhancers really are the really define the breadth of the gene because they are spread out so far across the gene that in fact actually coding sequence or the coding regions, the exons are rather small, but the enhancers are really quite spread out. Sometimes 50,000 base pairs away.
You knock out one enhancer site, it only affects one gene, but like, so transcription factors, for instance, if you have,
But if you knock out a protein that's a transcription factor, it can have its own effects on a whole bunch of other enhancer sites.
is it that there's only one, like, multiple transcription factors can bind to multiple enhancer sites, and enhancers can only bind to
I think, well,
if you knock out an enhancer site, it's only affecting that one specific, that's one. If you knock out a transcription factor, it's not gonna be able to bind and express one gene and also a different gene.
that's right. I mean, a transcription factor, if you knock out a protein that's a transcription factor, it's going to have lots of targets, unless it has one, but let's assume it has lots of targets, and all those targets are going to be affected in some way or another. What does it matter the name of the E1 protein? There are lots of, it's very rare that you have one transcription factor binding to one enhancer in the genome, but it does happen, yeah.
Isn't it possible for multiple genes to use the same enhancer?
So are you describing a situation where you have multiple genes on the chromosome,
Yeah.
and let's say this is a top strand gene, and you have the bottom strand gene like this, and they both share an enhancer.
Oh, so only if...
Is that what you're asking? No, I'm just saying, are you suggesting that?
No, I was just suggesting on the top strand by itself. On one strand, and two genes on one strand can use the same enhancer.
So you go into the fringe of, by the time you're sharing the same enhancer, are you sharing the same transcription start site? Are you sharing the same polyase? What else are you sharing?
Nothing.
But if you're sharing the common enhancer, then they are technically the same gene.
Okay.
Technically. All right, one last question.
Since they have multiple targets, if you are able to move the enhancer site to a different gene, would it still work?
So I'm not quite sure I understand the question. Let's say
Like E1, if you move that to like a different gene, like
E1 is a piece of DNA here.
if you're able to duplicate that,
Please, people, be a little quiet. I can't
You're
even
able
hear
to
the question.
duplicate the sequence for E1 in some other gene, would that gene be able to express in legs?
So this goes back to, this is the second part of our discussion today that I wanted to get to you from last week. I was drawing for you an example last week, Tuesday, of enhancers like this. And I said, what happens if you put Laxi with a minimal promoter, and that's it. So it's Laxi plus a minimal promoter. That means it can't express. But I said, if you're near an enhancer, you can, and I used the word steal last week, this week I'm going to use the word borrow. Because it seems steal is how we figuratively refer to it, but we're going to say borrow this time. You can borrow the use of this enhancer and activate this Laxi expression. And that means, as you properly suggest, if we now take this chunk of DNA out of the cell and we put it somewhere else, if we now take this Laxi with the minimal promoter and we take the neighboring sequence and we put it somewhere else in the genome, it too will function in the exact same way. Does that answer your question?
We're saying that the minimal promoter without an enhancer is effectively no.
That's right.
Expression. There would be some extremely low level, right?
Rare. So rare that, and they're very good about, for these particular enhancer traps, these are called enhancer traps, by the way. As it sounds, you're trapping enhancers. They are rigged to be extremely low, very little noise. Very, very low noise. So again, what I want you to get through is that you're borrowing this enhancer, that you haven't done anything technically to this gene as long as you haven't disrupted its transcription. This transcription factor will bind here and will attract RNA polymerase to this and RNA polymerase to this. However, some of you noted that if your other gene is too close to this transcription unit, you can start messing up this transcription unit. Then that's not a silent mutation. You've actually caused a defect to this gene. So they purposely try to identify reporters that do not affect endogenous genes. That means they can homozygose them, and they're not lethal, and they have no phenotypes.
The borrowing of enhancers is within the same cell, it's just in a nearby gene, right?
It's in every cell of the body.
But I mean, like, so if you have, like on the quiz where you have some cells that express some proteins and some that don't express all those proteins, the nearby cells can borrow enhancer proteins from other cells.
Normally, transcription factors are nuclear bound,
Right.
so they are not free to diffuse between cells. That's correct. Normally. So, so it's important you realize that these minimal reporter lines are able to borrow enhancers anywhere in the genome. And I told you, if this number of nucleotides is always somewhere around 6 to 8 nucleotides, that means 6 to 8 nucleotides is not hard to find even in heterochromatin, the non-expressed portions of the genome. Those are not hard to find, and thus, in fact, you don't always get these reporter lines to actually be near any gene. There happen to be near some 6 nucleotide sequence that wasn't able to drive RNA Pol II to bind to anything of relevance. Right? You need to have, so for example, let's think about this as heterochromatin. No genes. There are no genes in here. There are lots of nucleotides in here. Whole bunch of A's, T's, G's, and C's. Somewhere in them, let's say hypothetically, there's a TAA, TTG. That is a 1 in 4,000 chance of happening. And then let's say a few nucleotides away, let's say there's a TATA. That's a 1 in 256 chance of happening. And taken together, the probability you find those two just by chance is extremely high. It's really quite high. Because you're not asking it to be a particular spacing, so really the likelihood that you find this sequence next to this one is virtually the same rate. So TATA is a common sequence in the genome. So most likely, wherever there is this particular restriction site, you can find within close proximity a TBP binding site. But there are no genes being expressed. And why are there no genes being expressed? Well, it's not just that. It's just if you are going to initiate and make an RNA, that RNA has to be polyadenylated. That RNA has to be stable. That RNA has to leave the cell. There are lots of things that keep that RNA from actually ever doing anything. But you actually get these benign transcripts all over your genome all the time. You can have internal specific expression of CREF all the time. And again, that's why be careful what you wish for, that you think everything in the genome is coding for something of relevance. There's lots of CREF all over the genome that's being expressed in all kinds of tissues. So if you happen, for example, to drop in a LAC-Z reporter here with a minimal promoter, you might actually borrow this enhancer where there was never any gene. Yes?
I'm confused about how you reconcile the idea of
How I reconcile that?
how one
Okay.
of the idea of borrowing an enhancer from another gene and what you said before about each gene having their own enhancer.
borrowing a shirt from someone.
So, I mean, I think, so let's say, for example, I have two genes on my genome here, okay? This one is expressed in the leg, and this one is expressed in the antenna. They're next to each other, hypothetically. And we're going to call here E1, E2, and here we're going to say E2, and you need E1 and E2 to get leg expression, and you need E2 to get antennal expression. Okay? So, if I knock out E1, and this is actually a really good question, but I've never asked you on a quiz. I think this is much harder than a quiz question should be. If I knock out E1, are there any enhancers for me to borrow to get me expression? Let's call this gene X and gene Y. So leg expression of X, antennal expression of Y. And I knock out E1 in gene X. Are there any enhancers for me to borrow? I'm giving you this sequence here. Are there any enhancers for you to borrow to drive leg expression? But I'm telling you, the only one that's, I've scanned this whole thing. The only E1-like thing is here. I mean, that's implicit here, right? If I haven't drawn E1 here, it means it doesn't exist. Right? There's no point in me, why would I say that it's here if it doesn't exist? It just doesn't exist. You know, maybe this is our shorthand for speaking, but I'm not going to say no E1 site. I'm not going to do that. It's implied. If you didn't draw it, it's not there. Yeah?
When you have multiple copies of answers, just inseminate multiple copies.
So, but let me just start with something very basic. All right, so given this particular gene architecture, okay, you all agree that if we KO, we delete out the 6 nucleotides, we no longer get X expression. We're all on the same page there. What happens if we KO E2 here in Y? You might still have expression, because this enhancer, while far away, might still be able to coordinate with this transcription site to get it to work. Might. You don't know. And that's the hard part of understanding how transcription works for any given gene, especially if they're close to each other. And again, going back to his question, what if we had this in the genome? How do you know when you delete it here that it was not important because you could have gotten rescued from something a little further away? Absolutely happens. And it's very, very hard to define sites that way. And typically what people do is, going back to what he said, is if you really want to analyze this, you extract this from its place in the genome. You put it somewhere else in another genome and you say, does this work? Does it work like it normally works? And then you go and you delete it. And you do it in 3 or 4 different genomic contexts, so that you know that you're not borrowing an E1 enhancer by chance from some neighboring sequence. It's work. It's a lot of work to do to define these, but that's how they were all defined, through lots and lots of work.
If you do have the E2 knockout for Y, would you expect it to express at a lower rate than borrowing from X?
You don't know. You don't know. It all depends on your ability to borrow this sequence. The distance, how well it can and work with the minimal promoter that was here.
Sure.
You don't know.
So in theory, it could be just as good as.
Could be just as good. You have no idea that. In fact, you get fooled to think, I guess this sequence had no value. It's even worse than that. But in fact, this is part of the gene. This is not part of the gene. You're being fooled by what you borrowed.
Just one thing. Would gene X be expressed in the
No, because gene X so if you mean if you knock this out. In this particular configuration, maybe because there's no E1 expression. If you knock it out, maybe something bound here. I'll have to think about it, but maybe.
Do you get lower expression when you borrow enhancers, or is it essentially the same amount?
You can get the same level of expression if you borrow enhancers or lower. It's unknown. It's all empirically driven.
So if you would get the same amount using the borrowed enhancer, how can you definitively say that I mean, you just said that that is part of the gene, but first E2 is not part of the gene. How do we know that once you're using that E2 all along?
Because what you do is you extract it from the genome. And that's how you first begin to test. The first step in trying to figure out what the enhancers are in a gene is you extract it from its location and you put it somewhere else. Because as I told you, it's huge, the region. It's extremely big and you never know. And I mean, it's even worse than that. I mean, right, you think that it's regulating this, so you knock this out, you don't lose Y, but in fact there was a neighboring gene that also lost its expression, and it's that other gene which causes your phenotype. Think about that with medical genetics. You make a knockout, you lose functioning of Y, and you go, oh, it's Y because it's buried in the middle of the Y gene. It's actually the next gene over which caused the phenotype. Y has no phenotype at all.
So in theory, the whole time it might have been using one or the other E2. We can't say unless we've removed it.
That's right. And in fact, if you realize this is a great evolutionary trick to make sure you don't lose expression of Y. Right? If you're able to delete it here and still get expression, it gives you a lot more play to tolerate mutations. So this is not uncommon that you constantly borrow enhancers and stay working. And that's probably in part why all the genes that are expressed are clustered. So you have these bits of genomic region where genes are heavily clustered, and you have bits of genomic where there's absolutely no genes at all. All right, see you Tuesday.